So the next speaker is Javier Trepat from the IBEC in Barcelona, and the title of his talk is Mechanics of Cell Migration, Division, and Folding in Epithelial Sheets.
All right, thanks, Thomas, for the invitation. My title is overambitious. I will not tell you about migration and division. I will tell you mostly, only actually about folding. So everything that I will tell you about is unpublished work and still pretty far from being published. Okay, I don't think I need to introduce epithelial tissues after the talk that we have already heard, but just to remind you that these sort of layers of cells that cover the internal and external surfaces of your body are important, and the mechanobiology of these layers is actually determining a number of functions, such as tissue compartmentalization, protection against pathogens, morphogenesis, secretion and transport, wound healing, and tumor progression. So this has led several labs, including mine, to understand the mechanics of epithelial tissues. And I will just show you a few examples of that. So this is some old work now from my lab, where we managed to map the forces, the traction forces, so forces that cells exert on their substrate as a wound is healing. So we were able to see how there is a transition between forces that are actually pointing away from the wound to forces that are pointing towards the wound. And there is a transition between cell crawling and purse string contraction mechanisms. And one fundamental property of epithelial mechanics is that many of the phenomena that we observe are emergent phenomena, in the sense that you cannot predict them from the observation of a single cell. And one of these phenomena is the detection of gradients, so chemical gradients or mechanical gradients, as you see here. They need, so cells often need cell-cell junctions in order to detect these gradients. So this is a case in which we put an epithelial patch on top of a gradient of stiffness in the substrate, and we saw that cells tend to move towards stiff rather than towards soft. When you break cell-cell junctions or you prevent cells from being able to transmit forces, all these phenomena go away and expansion becomes fully symmetric. So often in epithelial mechanics, you need cells to form junctions in order to be able to exhibit a given phenotype. There's also been great interest in the rheology of epithelial monolayers. This is work by Guillaume Charas's lab, where they did a very hard experiment, which is to hang an epithelial monolayer between two cantilevers. They managed to stretch this epithelial layer to measure its rheology and to measure its fracture properties. So these type of experiments and many others have led to the development of a number of theoretical papers. And for example, here I'm showing the vertex models, the 2D vertex models that several groups have pushed forward. So I think we're today in a position where we can actually compare and confront experiments and theory at a pretty deep level. So it's a very interesting time in epithelial mechanics. However, there is a problem, which is that current understanding of epithelial forces largely relies on 2D measurements. Now, many of our epithelial sheets, like the skin, with the exception of hair follicles, can be assumed as two-dimensional tissues, but there's many epithelia that are actually three-dimensional. And one example that I'm sure we'll hear about is the early Drosophila embryo, where you have this one layer thick epithelium that's enclosing fluid-filled lumen, or you can go to organoids, where you can, of course, see that these epithelia are actually not flat. Now, if you try to understand mechanics of three-dimensional epithelia, of course, you need to understand the same mechanical variables, or at least measure the same mechanical variables that you need to measure in 2D. So you need to understand the force that one cell exerts on its neighbor. You need to understand the force that one cell exerts on the matrix, if there's matrix. There's one additional variable, though, that doesn't play a role in 2D, but is very important in 3D, and this is the difference in pressure between the lumen and the outside medium. So if we want to understand mechanics in 3D, we need to be able to measure tensions and tractions in the system, but we also need to measure this pressure difference. And this turns out to be very hard to measure, and there's little known about it, and there's no invasive measure. I mean, everything that has been measured, and there's very little measured, has been in a very invasive way. So we decided to tackle this problem and try to understand mechanics of epithelia in 3D. So for that, we went a few decades back, and we dug out this paper that was published in Science in 1969 by Layton et al. Actually pastes the entire paper as sort of a tribute to the times where science papers had 1 column and a half, and 3 microscopy images and no supplementary material. Because, I mean, these guys discovered something that was actually a very good idea. And this one thing that they discovered is that when you see the monolayer of epithelial cells and you let them grow to confluence, they eventually form these blisters, which these guys called MDCK domes. And this is something that anyone who's worked with MDCK cells knows. If you grow them in confluence, you leave a flask accidentally in the incubator, these guys form 3-dimensional structures, and this was the subject of lots of studies in the 60s, 70s, and 80s, mostly about epithelial transport and polarity. So we decided to build up on this work. So the idea is that these structures form because epithelial are polarized, so they pump ions from their apical side to the basal side. So there's a growing concentration of ions here. This creates an osmotic pressure, which drives water flow into the lumen. So eventually, this layer delaminates because of the pressure difference, and you end up having these dimensional structures. So this delamination caused by ion pumping, actually, and by osmotic pressure differences. Okay, so we decided to take this system and sort of combine it with current technologies, or expand current technologies to be able to implement this system. So what I'll show you is work by a PhD student in my lab, Ernest Latorre. It's a very close collaboration with Sohan Kelly in the group of Marino Arroyo, who did all the theory that I will be telling you about. Okay, so the idea is that we take this soft PDMS gel. So this is a non-porous gel. They are 3 kilopascal in stiffness. Then we coat it with fibronectin, but we leave some gaps that are not coated, so cells cannot attach there. Now, if we attach, if we see MDCK monolayers, and there they grow everywhere except for the non-adherent areas, as you would expect. Now, if you wait long enough, they also manage to invade these non-adherent areas, and this has been studied here actually by the groups of Pascal Silverzon and Benoît Ladoux, trying to understand the mechanism by which cells are able to invade without forming adhesions with the substrate. Now, if you wait even longer, then what happens is that domes build exactly on top of these non-adherent areas. So this is an example here of a 15 by 15 dome micropattern, where you can see that everywhere where there's a non-adherent area, there is the formation of one of these domes. Now, these are confocal sections where you can see each one of these domes. And so this is the Z section along this line here, where you can see that everywhere where we had a non-adherent area, we have one dome. And we can have them like separated only by one single cell, actually. So the patterning actually works very well. So what's quite remarkable is that despite the fact that the base has the exact same geometry, just a circle of the same size, the height of these domes is actually quite variable. So the reason for that, I'll tell you in a minute. We can make them of any size, actually, so, and shape. So we can make them elliptical, we can make them rectangular, we can make stars. And now the reason why the shape is so heterogeneous is because they are very dynamic in time. So if you take one of these domes, you image it as a function of time. These guys are growing, they're shrinking, sometimes they're collapsing, they're oscillating. So it's a very rich dynamic. So when you take a snapshot of all these domes in parallel, then you see them in very different states. They are very dynamic. They pump ions, they grow, and then eventually sometimes they burst, sometimes they just slowly collapse. Okay, now the question is whether we can use this system to measure mechanics. And our idea was that if this soft PDMS was soft enough, and this pressure in the lumen was high enough, then there should be an indentation right under the dome. And by measuring this indentation, we should be able to compute the exact value of pressure inside this dome in a non-invasive way. So we did that. So we did that by sort of generalizing traction force microscopy to 3D, as others had done before us. So the idea is that we have this layer of beads at the interface between the cells and the gel. And we measure the three-dimensional deformation of these bits, of this layer of bits as a function of time, and from that we compute the three-dimensional force maps at this interface between the cells and the gel. All right, so here's the data. Now in yellow, you see the traction forces as you are used to see. So these the two-dimensional traction maps in X and Y. So under the dome, of course, there is no traction forces because these are shear forces that can only be generated by cells. But if there's no cells, there cannot be shear forces here. Now the first row of cells shows a systematic pattern of forces that are pointing outwards. And then after this couple of cell layers, we see forces that are sort of oriented in a random way. Color coding shows the three-dimensional forces, so the forces in Z. So blue means negative forces, which means indentation, and red are forces that are pointing outwards. So under the dome, as you would expect, we see a force that is indenting the gel. And in the first ring of cells, we see a force that is sort of tending to pull cells upwards. And it has to be like that because, of course, all this has to be balanced. So if you plot this from a side view, what you see is that indeed there is a pressure in the lumen that is indenting the gel, and then there's a force that is pulling cells upwards and outwards in the first ring of cells. So we can actually measure directly the pressure that is under this dome, which turns out to be in the order of hundreds of pascals. Okay, so now if we have access to pressure, then one question that one may ask is whether we can actually measure tension in this dome. And the answer to that question varies from yes to no. So it's a no if the shape of this is complicated. But it's a yes if the shape is simple. And it's a very clear yes if the shape is a spherical cap. And it turns out that for every dome that we measure, these are always spherical caps. So then all you have to do is apply Laplace's law. So I measure directly delta P, pressure. I can measure very easily the radius, and then I know the tension. Note that the fact that this is actually a spherical cap, it means that the tension is homogeneous in the entire dome. Okay. All right. And it turns out that the numbers that we get are on the order of 2 to 4 millinewtons per meter. So for the experts in cortex, this is close to the values that people measure for cortical tension. Okay, so then we did a number of control experiments to sort of reassure that we were doing the right thing. For example, you can treat these cells with this inhibitor of rho kinase that Sasha was telling us about. So immediately after adding this drug, pressure drops very significantly, as you can see from this map. Volume doesn't change immediately. Remember that for volume of these domes to change, you need flow, and flow is slow. So basically, volume increases, but it does so very, very slowly. So what this means is that as pressure changes, tension changes in parallel, just because of Laplace's law. Okay, so this is reassuring that you treat these cells with a rho kinase inhibitor, and tension decreases by a factor of 3 immediately after adding the drug. Okay, now what you can do next is, well, I have access to tension in these domes. I can measure the radius of these domes, but I can actually also measure the strain in each individual cell in these domes. So I can start plotting relationships such as tension versus strain. And this is shown here, where you have on the x-axis the nominal areal strain. So this is 100% means that you're doubling the area of the dome, tripling the area of the dome, quadrupling the area of the dome. And this is surface tension. And each color is a different dome. So as you can see, for the first 100% strain, what we're getting is a linear relationship. So we get a linear increase of tension with area. But then we reach a plateau. So after 100%, so after the first doubling, data becomes scattered, but we sort of get a plateau here. So what this means is that you stretch and stretch and stretch the dome, but you're not changing tension, which is a very interesting phenomenon. So we tried to understand this, and our first guess of why we would have a plateau here came from a hypothesis that involved sort of tissue rearrangements, or at least I would say tissue scale mechanics. So one possibility was rapid directed cell division triggered by stretch. So there's been a number of papers recently that showed that when you stretch a monolayer, there's an increase in cell division. We actually have one paper that's coming up in Nature Cell Biology along this direction. Another possibility is network rearrangement. So there could be T1 transitions that could explain why, even if you stretch the tissue, you don't get an increase in tension. Yet another possibility is that could be cell exchange between the dome and the surrounding tissue. So we could have cells that are actually moving up the dome, and this is what explains that you can stretch, like you can increase fourfold the area of these domes, and you don't get a change in tension. So to make a long story short, we quantified this very carefully, and in the time frame where we analyzed our data, there are no cell divisions, very little cell divisions. There are very little T1 transitions, and there is no cell exchange between the dome and the surrounding tissue. So this is not a tissue scale phenomenon, it's actually a single cell phenomenon. So the mechanics that explains this behavior has to be at the single cell level, not at the tissue scale level. So then we wrote a very simple model that actually captures very well this behavior. So the idea is that you can sort of draw this hexagonal pack of epithelial cells. And in the simplest configuration, you could imagine that this is characterized by an apicobasal surface tension and a lateral surface tension. And you can assume that this is the same for every cell in your epithelium. So then you can then go to a configuration where you're actually stretching this monolayer, and then the apicobasal area increases and the lateral area decreases, as you would expect. Now it turns out you can write in a very simple way the surface energy in the system, energy density, sorry, which is just the product of tension times the area, so apicobasal tension times the apicobasal area, and the lateral tension times the lateral area, right? Very simple. If you want to compute tension, then what you have to do is compute the partial derivative of this surface energy density by the aerial strain of the cell. And this gives you an expression that is actually analytical and very simple. And if you plot that expression, this is what you get. So you get an initial increase, and then you tend to a plateau. The fact that you get this shape is actually quite intuitive. When you start to stretch the system, you have two contributions, one coming from the apicobasal tension, one coming from the lateral tension. As you stretch the monolayer, because the dome is growing, then the area, the apicobasal area increases, and the lateral area decreases. So everything then becomes dominated by the apicobasal surface area. This is what you're tending to. Okay, so this seems to explain in a very simple analytical way this behavior. But there was one problem, and the problem was that when we looked carefully at these cells, and there is a dome that is progressively shrinking, the area of the cells in the dome was extremely heterogeneous. So you had some cells that are huge, and some cells that are really small, in very close proximity. So this is better illustrated here, where you see one of these domes that is shrinking. There's two cells here that are stretched. This orange guy is stretched 900%, so it increased its area by a tenfold, and this guy by ninefold. So these cells are really, really stretching like crazy. And there's other cells that these little guys here, the green one and the blue one, are stretching only 100% or 200%, whereas the dome strain is 200%. So the response is extremely heterogeneous, and this is something that was not captured by our model. So we had to explain not only the fact that we get this sort of tensional homeostasis or constant tension at very high strains, but we also had to explain that this material, if you wish, had very strong shape heterogeneity. And this is what led us to the notion of superelasticity, which is something that probably many of you have never heard about. And I had never heard about this before we started sort of scratching our heads trying to understand the mechanics of this system. So in terms of the superelasticity, is a very well-known phenomenon in the field of metallurgy, which I don't know anything about. But superelasticity is actually what allows a number of industrial materials to exist, such as these glasses that you can actually fold, or brackets, or stands. So these are, thank you. So these are materials that you can actually, and these are metals, metal alloys, that you can actually take, you can fold them, you can stretch them, and they always go back. So there's no plasticity whatsoever. Actually, I brought a superelastic material here that I stole it from my dentist, and I will pass it in case you want to. So you can do anything you want with this material. You see that it's a metal that is perfectly elastic, has no plasticity whatsoever. So the reason why plasticity happens in the system is because there is a phase transition at the molecular scale. So you take one bar of this material as the one that I'm passing. You can stretch at the beginning. What will happen is that you will sort of deform the atomic structure of this material, and you'll get a linear relationship between stress and strain. But if you keep stretching, what happens is that you have a phase transition at the microscale, at the nanoscale, actually. So you go from one phase that's called austenite to one other phase that's called martensite. So as you keep stretching, at the molecular scale, you get this phase transition that explains where you reach a flat stress-strain regime. So this is sort of a very oversimplified way, but the message is that actually a phase transition between a non-stretch and a super-stretch phase allows you to have tensional homeostasis in case of metals. And this is perfectly reversible in this, as in the metal that I'm passing. So we thought that this was actually very analogous to what we're finding experimentally. So we see this linear increase at the beginning and then a plateau, and we also see two phases, one phase that corresponds to cells that are super stretched, and another phase of cells that are not stretched or very weakly stretched. So this happens thanks to this microscopic mechanical instability. So what we had to figure out now is what would be the equivalent, what would be this mechanical instability that would explain the existence of these two phases in epithelia. And our model is the following. So if you take one cell and you stretch it, and the mechanics of this epithelial layer are largely determined by the cortex. Now, if you stretch it, the density of the cortex will actually remain constant. And this is because you have lots of G-actin in the cytoplasm, so as you increase the surface of this cell, there will be no problem from actin to polymerize and to maintain a constant thickness of the cortex. So these are the prevalent models that have developed and are actually experimentally investigated by a number of researchers. Now, if you keep stretching, though, we thought, well, maybe if you keep stretching this system, at some point you will run out of G-actin, so you will not be able, this cell will not be able to maintain a constant thickness in the cortex. So now if this happens, once a cell reaches this regime in which it cannot polymerize a cortex of constant thickness, then this cell will actually start softening. So then you reach a mechanical instability. So there is cortical dilution, not sufficient G-actin to polymerize the cortex. This will lead to softening of the cell. And if the cell is softer and the dome is under the same tension, then this cell will stretch more, which will further dilute the cortex, further softening. And so you will reach an instability that will cause this cell to become super stretched. So we measured if that was the case. And so for the first 100% of cell deformation, when we measured the density of actin density in the cortex, we found that there was no change as published before by others. But as these cells were becoming super stretched, then the cortical density actually decreases. And this is actually the data that support this quantification here. So indeed, these data support this idea that there's this phase transition caused by this mechanical instability between cells that are super stretched and cells that are not super stretched. Now, one question that you might have then is, well, if this cell then becomes super stretched, what prevents it from becoming even bigger, right? I mean, I showed you some cells that are that increase their area by 10-fold, but not 100-fold, right? So what limits the ability of these cells to become even more super stretched? And the several possibilities, that one that we're particularly excited about is intermediate filaments. So intermediate filaments in cells that are not stretched are sort of there in the cytoplasm. You come with a laser, you cut the intermediate filaments, and the shape of that cell doesn't change whatsoever. Now, in cells that are in these domes, and in particular the superstretched cells, we see that intermediate filaments are like tensed ropes, and you go with you come with a laser, you cut them, and then that cell becomes even bigger. So we think that intermediate filaments are the ones that prevent this further superstretching of these cells. Okay, so finally, the question that we asked is whether this is relevant or not. So we browsed the literature. We found this paper by Deglicenti et al. Looking at human embryos at day 14. And what they were able to show is that in the trophectoderm of this embryo, there is a very high heterogeneity, and you can hopefully see here some cells that are superstretched near some cells that are not stretched at all. So in tissues that might need to become highly stretched, this mechanism might allow them to really accommodate in a very reversible way, actually, this behavior. Okay, so in conclusions, we did these DOMS 2.0, which allowed us to control geometrically these MDCK folded structures. This gave us access to epithelial mechanics in 3D, and then we sort of uncovered the superelastic behavior of cells in which a mechanical instability leads to this coexistence of two stable phases, cells that are superstretched and cells that are not. In my title, I had the word division. I will show you just one slide about division. This is work that was recently accepted in Nature Cell Biology and will hopefully be soon out. So we monitored the expansion of a monolayer of cells expressing Fuchi for 48 hours. And then we asked the question, can mechanics predict the duration of the cell cycle? I will not tell you anything else about it, but I will be happy to discuss in case someone is interested. The answer is yes, actually, as you would have expected. Okay, so let me just thank people in the lab. So Ernest did most of the experimental work with these DOMs. And this has been a very nice collaboration with Marino Arroyo, who did all the theory that I told you about. We had some help from Benoît Ladoux for this micropatterning, and these are also collaborators in the lab, in my lab and in Marino's lab. Thank you for your attention.